Vai aparecer uma...
beleza.
Pronto.
Deixa eu ver aqui, o meu saiu. Pronto, tá aqui, entendi. Tá ótimo. Okay, so we're gonna start our talk today. And I have the great pleasure to introduce Cecília Cavazzoni. Sorry, Cecília, I think I vandalized your name. So Cecília is very dear to me because we shared a lab for a while. So she graduated from biological sciences, biomedicine, actually from Unifesp in 2011. And after that, she came to Rio. And over there, she was advised by Luiz Eduardo Coelho Andrade. And when she came to Rio, she started her master's with Alberto Nóbrega at the microbiology department here in UFRJ, where she graduated in 2014. And she met André Vale, who advised her through her PhD, which she graduated in 2018. And during that time, I met Cecília at the lab, and it was amazing how much she worked and her the upbringing, the upbeat that she is, and she's a very, very nice co-worker. During that time, she spent a year at Rockefeller working as a sandwich student at Gabriel Vitoria's lab. After that, she did a brief postdoc here at UFRJ before moving now to Boston, where she's doing her postdoctoral, she's a postdoctoral fellow at Peter Sage's lab at Brigham and Women's Hospital. Over that time, she has contributed to 15 papers, where she's the first author in 3 of them. And she has also, during this short amount of time already, helped advising 4 students, undergraduate students. And now she's giving us the pleasure to share the work that she's been doing during that time. So thank you again, Cecília, for accepting our invitation and for giving this talk to us. And so I'll pass you the word now. Thank you.
Thank you. I'm very happy. It's always good to be home. Feels like home being here with you. So it's always a pleasure. So yeah, as Miriam mentioned, I'm here now at the Sage Lab in Boston at Brigham Women's Hospital, which is a teaching hospital of Harvard Medical School. And I've been working in this lab for the past 3 years, a little bit more. So I'm going to show you some of the work we've been doing, some part of it that's a little bit more wrapped up already, and then a little bit more on new insights and where we're going with this story. So basically what we're studying in the lab is the crosstalk, the help and the regulations that happen within germinal centers. So that's why I chose this title of Follicular T cells and B cells cooperation in germinal centers. So we've been able to look more closely into how these 2 cell types talk and cooperate in germinal centers to optimize the humoral immune response. So we're looking mostly at the output of protective antibodies that come from the GC. So here I just wanted to first start with a real image of the structure that we're looking at. So here we have a close look in one B cell follicle, one germinal center that is highlighted in this box. So this is where the 2 B and T cells talk to then generate the protective antibodies that we need to be able to handle infections or respond to vaccination and then be protected. So some of you in Rio might be familiar with this slide, and I've seen even some updated versions of it going around there. So this is just to talk a little bit about the dynamics of the humoral immune response. So we do have many different components to it, and I just wanted to highlight the one we'll be focusing on today, but also give you an overview of, So we'll be focusing on this second wave today, but we do have natural antibodies which are constantly being produced, secreted, and are constantly circulating and give us a first line of defense. Then upon encounter with antigen or pathogen, we get a first wave of lower affinity antibodies that are quick, but are less tailored towards the antigen that has been seen in our body. And then we get this second wave that is dependent on T cell help, and that gives us higher affinity antibodies, class switched ones, and so on. So we'll be focusing on this part of the humoral immune response and how it's generated. And as you may be more or less familiar with the germinal-centered structure, I like to bring this slide just to give us an idea of how dynamic this structure is. So this is one germinal center schematic. So this is where the conversation starts when B and T cells interact, and this interaction is based on antigen recognition. So it depends on the two of them being able to see the same or to show each other the antigen. And then B cells get activated and they move to the dark zone where they proliferate and they hypermutate their B cell receptor, their BCR, and then they go back to the light zone where they meet T cells again to sort of check their BCR again. And that cycle repeats over and over until they finally exit as plasma cells or memory B cells. And the criteria here for them to exit are also still being better elucidated. We don't know exactly at what point these cells exit as one or the other, but there's some evidence, some working hypothesis. And so this is kind of what happens. The stronger the signal they get here, the more they proliferate, the more they hypermutate, the higher the chances of acquiring a mutation that gives them higher affinity, and then they burst. This is the clonal burst is the term that we use to say this B cell proliferates a lot and kind of takes over. So pretty much all the B cells within that germinal center are derived from this one that got a lot of T cell help and was able to proliferate a lot. So this is the structure we're looking at. And now I'm going to zoom in a little bit more even into the B cell receptor, just so that the data I'm going to show you makes sense. So this is the structure of the BCR and the heavy chain. And as you know, there is a variable region in antibodies that confers the specificity. And within the variable region, there are more variable regions. And so we'll be looking at a lot the most variable of the variable regions, which is the complementary determining region 3 in the heavy chain. So it's called CDRH3. And this region is the most variable because it comprises the junction of different gene segments that form the BCR. And there's also room in there for some insertions or deletions that are not germline encoded. They're not encoded in the DNA, genomic DNA. So that's where the most variability is generated. And so we use this region to define clones to identify the B cells and their descendants. And I'll be showing you a lot of data on mutations that occur in this variable region. So that's why I just wanted to bring this slide so that we are on the same page as to how do we look into the B cell evolution. And so now I'm going to introduce you a few other players in this picture. So within the B cell follicle, as I just mentioned, we have TFH cells that are T follicular helper cells, the ones that are able to crosstalk and activate the germinal center B cells. And more recently, we learned that we also have regulatory cells. So we have T follicular regulatory cells that are also within, outside and within the follicle. And their function seems to be suppressive. So they kind of balance out the activation that TFH cells give to B cells. And in this manner, they can prevent, or actually, I like to say it the other way around. They favor the ones that are the B cells that are more tailored towards the response, and they kind of dampen the ones that are not so effective or related. And in this manner, they also prevent the generation of autoreactive antibodies because they're not as fully targeted to the foreign antigens, so they get suppressed. So in our lab, we kind of look into all these three players that are in the B cell follicle and how they get there and how they exit and kind of their life story. And it's not clear yet if TFR cells only interact with TFH or also with GCB or how this suppression happens, but we are getting more and more knowledge on how this occurs and how we could also benefit from it. Sometimes removing regulation could be beneficial, sometimes boosting this regulation could be beneficial. So if we understand that better, maybe we can improve vaccination or help people with autoimmune diseases and so on. So we'll be talking about all these three players, and I'm going to start with the TFH cell part of the story. There has been some TFH cells weren't discovered too long ago, so there's been some evolution on the understanding of these cell types and how they get activated, how they gain access to the B cell follicle, and what are the requirements for that to happen. So this is how first they were kind of divided into pre-TFH and GC-TFH before and after they reach the B cell follicle. And one important molecule for them to migrate towards B cells is CXCR5. So it became a marker of how to define these cells. So the GC-TFH were defined as CXCR5 high, PD-1 high. And from our studies and others, we know that now that there's a lot more involved, there are other steps, there are different phenotypes that they can look in these two anatomical locations, they can have different phenotypes. And so we're also working on that in the lab, but I'm not going to show you that today. I'll just show you the actual GC B and T cell interaction data. So we first started, when I first moved here, COVID happened. So we started looking at these crosstalk of B and T cells in the context of a SARS-CoV-2 spike protein vaccine. So the first step was to determine how good of a response that vaccine would give us. So we started vaccinating mice with the spike trimer stabilized and MF59, which is similar to Adavax that's approved for human use. So we started to actually, Adavax is the one we use that's similar to MF59 that can be used in humans. And then we looked at the germinal center response to these vaccines first, just in wild type mice, just to get an idea of how good of a response we would get. And as you can see, it wasn't crazy good. It didn't elicit a gigantic germinal center response, but it was there. So we could see the germinal center formation in vaccinated mice. And here you can see that we'll switch PD-1 positive or or ICOS positive, CXCR5 positive to define TFH cells. They can be defined by either one, ICOS CXCR5 or PD-1 CXCR5. So they kind of have the same look if you take them by one or the other. ICOS is upregulated first, so you might be getting some more recently activated ones here. So as you can see, we could see the formation of TFH cells. We didn't see too much of an increase in TFR cells at this point, but this is day 10 after vaccination. But we did see formation of germinal centers, as you can see here, and also the appearance of spike-specific antibody in serum. So we did have a model for to study this vaccination in mice. So we went on with this, and we used this technique, which you might also be familiar because we started doing this in André's lab in Rio. So, and they still do it and I still do it here. So, and it's a great technique to study GCB cells. So what we do is to sort single cells into a 96-well plate, and we culture them individually and they proliferate. So we use feeder cells that give them CD40 ligand signals, BAF and IL-21. So they survive, they proliferate, and they secrete antibody in culture. And then we can screen those antibodies for the vaccine specificity. We could even do some affinity measurements that I'm going to show you. And then we can also harvest the cells at the end of the culture and sequence them. So we can get the information of the sequence and the binding properties of the antibody from one single cell culture assay. So this is what we did here. And this is just one example of B cells that we sequenced in wild-type mice. So as you can see here, at day 14, about a quarter of all cells in the germinal center are IgG cells in the germinal center are spike-specific. And we saw that with Zika virus as well and different antigens. That seems to be a pattern that early on, around this time point, 25 to 50% of the cells are antigen specific. And then we broke down this spike specificity into epitopes because we knew and we know, right, that RBD specific antibodies tend to be the best neutralizers, although there are exceptions of other targets that are also very good at neutralizing the virus. But just to have an idea of potentially protective ones, we split them in this 3 domains. So S1 here means S1 not RBD, because RBD is part of S1, but there are more epitopes in S1. So when you see S1 here, that means not RBD. And then S2 and not determined. So sometimes when you break down the protein, the binding is no longer there. So it's either a conformational epitope or it then becomes low affinity, and then you cannot measure by ELISA. So those are the non-determined ones, the NDs here. And here's some that we sequenced. So we got some information on how spike-specific clones were formed. And then here we also even measured the affinity of some of them. So we could correlate the sequence with the specificity epitope in the, not exactly epitope, but in the subdomain level, and affinity measurements. Okay, so we wanted to see to study more deeply the role of TFH cells in this whole phenomenon. We know that TFH cell help is important, but we wanted to learn step by step what was the requirement, at what point, and what are the consequences of the T cell help to a B cell. So we developed in the lab this model that is able to deplete TFH cells, also TFR cells, I have to say. Although we call it TFH deleter just for the sake of simplicity, but it is a CD4 so it actually deletes TFR cells as well. And so this is like a dual gene requirement for the deletion. So the mouse is CD4 CXCR5 log stop logs DTR. So every cell that expresses CD4 and CXCR5 will have DTR on the surface. And so when we give diphtheria toxin to the mouse, that depletes all the cells that have the diphtheria toxin receptor on their surface, which in this case means TFH and TFR cells. And this was published in a transplant paper. So we also studied transplant in the lab. So we first used this model in a transplant rejection study, but then we decided to use it as well in vaccination and see the effects. And the good thing here is we have temporal resolution. So we can give the mouse DT at a certain time point. So we can play around with having TFH cells early on or later and so on. So this is just to show how the model works. So these would be the TFH gate. And as you can see in the deleters, when we give DT, the TFH population is very much reduced. It's not completely gone, but it's very much reduced. As you can see quantified here. So as I mentioned, it does deplete TFH and TFR cells. But since we have data in a lot of people that the role of TFR cells kind of goes through TFH cells, so we think that in this model is not, we can still assess the role of TFH cells, because even if TFR cells were there, they wouldn't be able to suppress TFH cells, and their effects would also be absent anyway. So this is what we did. So we used the TFH deleter mouse to vaccinated them with the spike protein vaccine. And then for this first round of experiments, we gave the mice diphtheria toxin early on. So from day 2 until 11, we don't do this every day. It's every other day or every 3 days. So 3 or 4 doses of DT, because the mice also develop antibodies against DT. So repeated doses are a problem because they started to neutralize the toxin. So it has to be an acute treatment. So we did this, and we deleted TFH cells pretty early on. And as you can see here, the deletion was effective. They came down to 1/3 or less of the total TFH cells. And it doesn't affect, as we expected, these are just for controls, doesn't affect CXCR5 negative populations or B cells. But as you can see here, it does reduce the frequency of germinal center cells also by 1/2 or 1/3. And then it also, here's a little bit hard to see, but we did see a little bit of a reduction in serum antibodies that were RBD specific, not in total spike. But also at day 14 is a pretty early time point to see GC-derived antibodies in the serum. So these could be just from the first initial response, and not all of these are probably germinal center derived. So, but we could see that the deletion of TFH cells reduced the frequency of germinal center B cells and RBD specific IgG in serum. And so we did the single cell cultures to take a closer look at the B cell dynamics within the GC and clonality and so on. So what we found was, as we had seen before with Zika virus and other proteins, that about 40% of the cells in the control mice were specific to the spike protein, whereas in the deleter, there were only about 12%. So there was a reduction in antigen-specific germinal center B cells. And then also, as you can see here, most of these, or about half in the control mice, were S1 or RBD specific, whereas in the deleter mouse, we didn't find any S1 specific clones, and some were RBD, but I didn't show here the ELISA data, but it seemed lower affinity. So we went on to actually measure the affinity of these antibodies by biolayer interferometry. And indeed here, the KG is inverse correlates with the affinity. So the lower it is, the higher the affinity. So as you can see here in the control mice, there were some very high affinity antibodies, whereas in the deleter, we kind of found this dual behavior. Some were very good antibodies and some were pretty bad in terms of affinity. So we can hypothesize this. Probably what's happening is some cells are getting a limited amount of help and are still able to evolve into high affinity antibodies, but some are being left with no help and they are producing some pretty low affinity antibodies. So, and then we went on to sequence these cells and analyze the somatic hypermutation patterns in this BCRs. And so, as you can see here, when we split the clones by, so here singleton means the clone that we found only once, and then expanded means one clone or a B cell clone that we found multiple times in the data set. And so especially, so these are the greatest, the most likely the ones that are being selected and evolving. So they're dividing and expanding. And so especially when we look at these, you can see that in the controls, the distribution of mutations peaks at a higher number than in the deleters. So they're probably being able to get less help in the TFH deleter, so they're unable to mutate as much as the control. And we can also quantify this, dividing them in single clones, middle, we could say mildly expanded, and very much. So the ones that we found 4 times or more in the data set. And here's where the difference is more pronounced. And these are more clearly less mutated than in the control. So basically, we were able to assess with this model the contribution of the TFH cells to the clonal evolution of B cells within the germinal center. And then we can also do this, this measurement of clonal diversity, which I learned in Gabriel Victoria's lab. Is one way of quantifying what's going on in the germinal center. So this is, it's a measurement of how many clones account for 75% of the total. So the more expanded clones you have, the less number of clones you need to make up the 75% of the data set. And so as you can see here in the deleter, we have less clones being necessary to make up for 75% of the data set, which means lower diversity. So we have a few clones that are expanding a lot and kind of taking over, probably the ones that are still able to interact with the few TFH cells that are there. And then in the control mice, where you have a normal amount of TFH cells that are able to help a greater variety of B cells. And so you get more diversity overall in the germinal center. So this is kind of how we graphically summarized it. So in a normal condition, so both are good and bad is complicated, right? This terminology, but having diversity is necessary, but being able to select the best ones is also necessary. So we need this balance between keeping diversity and being able to select the most effective clones, right? So not having enough diversity can be a problem, and not be able to select the best ones can also be a problem. So in the normal condition, we kind of have TFH cells and B cells in a certain amount that gives us that balance. So we have a medium or normal, we could say, clonal diversity, and we have affinity maturation. Whereas when we don't have TFH help, we have a lower clonal diversity and therefore a lower affinity maturation, although some clones were still able to evolve and develop a high affinity BCR. And so then, as I mentioned, we decided to also look into the role of TFR cells in this balance. And so we have another mouse model in the lab for that, which is the same principle as the CXCR5 lock-stop-lock GTR. But in this case, it's associated with a FOXP3 So every FOXP3 expressing cell that also expresses CXCR5, which are TFR cells, will have the diphtheria toxin receptor on their surface, and therefore they can be depleted by diphtheria toxin administration. And so this is just data from the paper where they first published it, and they saw that TFR cells regulated vaccine-specific responses. They saw an increase in IgE in allergy, more formation of autoantibodies without TFR cells. So there was a previous publication in the lab on the possible role of TFR cells in different contexts. So we also used this mouse with the spike protein vaccine. And so as you can see here, it was the same regimen of early on GT administration. And we can see the depletion of TFR cells without affecting TFH cells or other T cell types. And as you can see here, as they observed before, there's a higher frequency of GCB cells after vaccination when you remove TFR cells. They're more class switched as well. And this didn't reflect yet in more antibodies in the serum, as you can see here. And then we also looked into the germinal center clonality by single cell cultures. And again, this looks very similar to the lack of TFH help, if you remember from the previous experiments. About 25 to 50% of the cells in the control mouse are specific. Many of them are RBD or S1 specific, whereas in the TFR deleter, even though the GC frequency was higher, the frequency of spike-specific cells is lower. And a lot of them, we could not determine the epitope, either because it's a different epitope or conformational, or when we split the domains, we cannot see, or because they're very low affinity, and then we cannot assess that by ELISA, which we can show here is the case for some of them. So here we measured the affinities by bilinear interferometry, and you can see a very similar pattern to the TFH depletion, right? So a lot of the clones are very low affinity. There are still some able to acquire high affinity, but for the most part, they are lower affinity than the controls. So we could show that the TFRs are also necessary for the presence of spike-specific cells in the GC and the affinity maturation process. And again, analyzing mutations, we can see the same thing, that the expanded clones in the deleter mice are less mutated, which was also quantified here. And in terms of diversity, there was not a huge difference like in the TFH deleter. They're still pretty diverse, but apart from this one very expanded clone in one germinal center, but overall diversity is similar, but the ability to focus on the antigen, the vaccine antigen, was impaired. So this is kind of how we graphed it. So here is the situation where we have less TFR cells to kind of put a bottleneck in the germinal center. So you get more B cells to participate in it, but then they're unable to get appropriate TFH help. And that also compromises the affinity maturation process. So this was an unappreciated role of TFR cells, but we were able to show that they also contribute to, they're not only suppressing TFH cells, but that, however, the mechanism, they are contributing to the affinity maturation process. So our idea is that they are putting a certain bottleneck, which we don't know the molecular mechanisms of how this happens, but it seems like they're putting a bottleneck in how many B cells can enter the germinal center and or participate in the reaction to balance that, to make sure that they can get the appropriate amount of TFH help. Okay, and then we thought about this one context where TFH, TFR, and B cells are already naturally altered, which is in aging. So a few years ago, Peter Sage himself had this paper on TFH and TFR cells in aging, and he showed that there's an increase in TFH and TFR cells with aging, but their function is impaired. So although there are more cells, they're not as effective. And there are other publications as well on the B cell side that with aging, B cells also become less responsive. So we decided to look into these balance of TFH, TFR cells and the germinal center in aged mice as well. And so as you can see here, we confirmed that these mice, our aged mice, had these phenotype. They had more TFH cells, more TFR cells, but less GCs and less antigen-specific antibodies in the serum after immunization. So this is 14 days after immunization. You can see the young control. Young means adult. It's not a pup, but the adult mouse that is the same control in every experiment compared to the aged one. And the aged mice were very much impaired in their ability to generate vaccine-specific antibodies. So this kind of the scenario where we wanted to look into the GCs more deeply to understand what's going on in the germinal center of an aged mouse. So we did the same thing, the single cell cultures, to look into clonality and clonal evolution within the GC. And to our surprise, this to me was one of the most surprising and pleasing findings. In germinal centers of aged mice, there's still a pretty good selection process happening. So although there are less cells, they're still able to focus on the antigen-specific cells. So for the most part, just like in the control mouse, many of these cells were spike-specific, and many of them were S1 or RBD specific. So even though there are less cells, they're still able to, so maybe the increase in TFH and TFR cells together are still able to keep that optimal balance so that the GC can still select antigen-specific B cells. And then for some reason that we also still don't understand, this doesn't translate into an optimal serum antibody response, right? So there might be some plasma cell differentiation impairment or other things. But what we could tell is that maybe we can help boost the function of these cells, and they're still able to select properly. And when we looked at the mutation pattern, we saw this also unexpected or maybe expected, but interesting to see. We have this bimodal pattern where there's a lot more cells that do not acquire mutations. There's still germline in their sequence, but there are also a lot more that are very highly mutated. So these, as we know, could be derived from more memory cells. We know that over time, all the B cells tend to be have accumulated mutations already, so the starting point is not 0. So we did observe this dual behavior here. A lot of cells got help, but were not mutated, and many more than the controls, young controls, had a lot more mutations as well. In terms of diversity, we saw the same, no difference in diversity. Both of young and aged were able to select and expand spike-specific clones. So that, I think, was pleasing and got me excited about these results, because then we might be able to figure out a way to make vaccines more effective for the elderly population, which is still a problem, right, that we need to address. So this is the full story, kind of everything we assessed in terms of TFH and TFR function, and then one context where they're both altered, but in some way they still enable optimal selection within the germinal center. So in aging, there are more TFR cells, so the bottleneck might be more stringent, and then we have less GCB cells, but that in a certain way maybe guarantees that all of them will get TFH help and then be able to hypermutate and evolve and affinity mature towards the antigen that we vaccinated the mice with. And then, as you know, we also, one great feature of the adaptive immune system is memory, right? So we know that once we're exposed to a certain antigen, we get a faster and more effective response towards the same antigen or a similar one once we encounter it again. And this paper was, I participated on it from Gabriel Victoria's lab, where we were looking at the memory B cell compartment and its contribution to a secondary response. And what we saw, which was pretty surprising, is that actually very few memory B cells make it into the GC again. For the most part, they become plasma cells right away and secrete antibodies. So for the B cells, it's more rare to get into the GC again and re-diversify than to, once you've been selected as a good binder to a certain antigen, you're more likely to become a plasma cell right away than to go back to the germinal center. And Gabriel's been working on it to understand what's selecting these cells to come back or not, and so on. So we decided to look into that as well on the T cell side. So basically what we did was secondary responses, where we deleted the TFH cells in the primary or secondary response, and then looked after a boost what happened. And so here I'm going to go through a little bit faster, otherwise we won't have much time to discuss. But what we did was to use the TFH deleter, delete the TFH cells in the primary response, and then 30 days later, give the mouse the same vaccine again. And as you can see, the titers of antibody in the serum go through the roof compared to the primary response. There was no difference in TFH or GCB cells at this point. So after the DT treatment, TFH cells were able to differentiate again later on. So this looks kind of a normal secondary response compared to the control, and they're able to produce a lot more antibodies. And here we have a log scale, so it's hard to appreciate, but we did see a bit higher titers of spike-specific antibodies after the boost. And what we think is because we made the selection more stringent in the primary response by reducing TFH cells, we got a better or faster secondary response. And so looking within the GC, that was more or less what we saw, that kind of dual pattern again. So more or less normal frequency of spike-specific cells within the GCs, but in terms of affinity, we still had some lower affinity cells or BCR in the secondary response. So they might derive from precursors that were unable to acquire higher affinity, or those cells just didn't make it into the GC again, and they're starting over compared to the control. And here's the same thing. So we did see less mutated expanded clones, even deleting just in the primary response. So it did have an effect in the memory compartment as well. So this kind of our hypothesis to explain what we saw is maybe a poor primary response also compromised the secondary response. And then we did the same thing with the TFR deleter. And then again, in the secondary response, they look normal again. So there's no more effect of GT. But in this case, we saw lower serum antibodies at day 8 after boost, which might be, again, because of the formation of less high affinity precursors in the primary. But then with TFR deletion during primary, we got pretty normal germinal center response in the secondary, both in terms of affinity and of specificity of the clones in there. So what we think, or we know that TFR cells have their important role is early on in the GC, mostly in the recruitment phase. So maybe that TFR deletion in the primary response did not compromise so much the secondary. But we still did see difference in the most expanded clones being less mutated. So we did alter the memory compartment somehow as well. So again, we're showing what was not appreciated before, that TFR cells also contribute to this balance that leads to optimal selection in the GC. So they're also important and necessary to an optimal antibody response. So here kind of we went through this already, all the summary of what we saw. And then just to give you a little bit of what we're working on now, recently it's also being shown by Julia from Michel Nussenswag lab that there's also evolution or clonal expansion and contraction in the TFH compartment. And so what we're looking at in the lab right now is more of the what's next for TFH cells. So how they become a TFH cell, and then what happens to them after as well. So as you can see here, there are all these factors to be considered in CD4 T cell memory right now. So this is a very recent review, and it kind of gives us an idea of anything can happen, apparently, to a CD4 T cell. So what we're trying to understand is, once they become a TFH cell in a lymph node or in a tissue, what happens to them and how they contribute to future responses, if they do, or what happens. So this is kind of what we are currently working on in the lab. Okay, and with that, I want to thank everyone in my lab, especially Peter Sage and Manuel, who's this other postdoc and who's been heading this project with me, and all the other present and past members of the lab and all of our collaborators. And the Sage lab is growing, so if you're interested, check out our website or get in touch with me, and I'll be happy to give you more information on it. And yeah, thank you all for your patience, and I can take any questions.
Okay, I think now it's working. Thank you very much, Cecília. Sorry over here. Thank you for the great talk. We have a tradition that we usually have the students asking questions first, if there are any. I'm not sure if there are any in São Paulo, but if not, then I can start to break the ice a little bit. Mm-hmm. But okay, sorry to be on the side. I try to. Okay, we have some deal with the camera over here. It's too smart for me, I would say. So I thought it was really interesting, everything that you showed, and it's amazing how you can change the aspects of the antibodies that you're generating depending on the function of the regulatory cells, and how you have this balance, right? And I think I have two questions. One is more conceptual, so I'll leave it to the end, but the other one would be, I think struck me the difference that you have between the specificity to the S1 and the RDB. Mm-hmm. And it seems that in the wild type mouse, you have both. You have antibodies against both showing up, but if you take out both the TFH and the TFR, you have a switch, and you have our anti-RDB, but you don't have anti-S1, and mm-hmm. It changes afterwards. And then more strikingly, when you do the recall response, when you try to do the recall response afterwards, in the 30 days after, and at least in the TFR deficient mouse, or the deleted mouse, let's say, you get the opposite again. You get more anti-RDB, and you have less anti-S1. So I wonder, does that matter? Like, does it make a big impact? Are you discussing this type of a specificity of how you can choose to have more anti one end or the other one, and how you're dealing with that, if it's a topic that you're studying?
Mm-hmm. Yeah, so our idea when we started characterizing them with that division of specificities was to do protective assays. So we wanted to do either neutralization by flow, or we were hoping to get the actual virus that we could infect mice with and check for protection. So that has just, for us, it kind of worked as a surrogate of potentially protective antibodies at first. Then we actually never got approval or the structure to get the virus here and work with it to infect the mice and see protection. But that was kind of our thinking when we decided to look into RBD or S1 antibodies. So if we consider that RBD are potentially more protective, what we're looking at, our optimal response would be the more RBD, the better. So that was the thinking. But we cannot be sure that that is the case. Like, we were not able to do actually protection assays or infection assays to make sure that that is the case. It was just based on the literature. Most of the neutralizing antibodies that people were describing were RBD specific. and also is one of the antigens that the immune system gets to see the most in the viral structure, right? So, but also our spike trimer was stabilized, so it didn't have the conformational changes that naturally happen during infection. So there are a few caveats there to, to actually say this mimics protective antibodies, or we could take these as a surrogate for protection. So, but that was the thinking when we started, yeah.
Okay. And this more conceptual question, I think, is wonder something that you discussed in the lab or even in the field when you're evaluating the old versus the young mice, and you look at their repertoire. I wonder how, because these mice are SPF, right?
Yeah.
Therefore, there might have encountered very fewer antigens than anybody would find. And so do you, are you doing something similar? Can you in humans, or are you trying to put these mice in an environment where they would have a little bit more of a diversity in terms of life experience to see how that would impact, and how does that play out in terms of the repertoire that these mice would have?
Yeah, right. Yeah, that is we did think about it, but we're not currently addressing it experimentally. What we're doing now is trying to understand what's in the environment of an aged lymph node that is causing this to happen. So we're still working on aging, but mostly on the side of what's there in the environment that is impairing the function of TFH and TFR and B cells. But so as we know in humans and even in more exposed mice, towards the end of our lives, pretty much every cell is a memory cell that has seen something, right? But we did not address that, or are not currently. Yeah, but we would love to, but we're not currently doing it. Yeah.
Yeah, because I found it impressive that there are so many cells that had little mutations, right? And I wonder maybe if you did several protocols of immunization in the lab, such that you didn't have to expose something dirty, but maybe immunize them against several different things of albumin, and then check for the specific ones for SARS-CoV-2 if that would make any changes or differences. So thank you. In the chat, there is a question from Gabriela Maciel. Did you look at the fibrosis processes in the aging? I'm just thinking about one more piece of this puzzle.
Mm-hmm. Right. Yeah, you're right, Gabi. We did not look into that, but we are planning on looking at the entire architecture of an aged lymph node. So we are gonna look into stromal cells as well, and some of that, and how that affects the dynamics of the GC response. So not fibrosis per se, but what else is in the environment that is causing this to happen. Yeah.
Okay, Bianca Braga is saying congrats. I think we all missed you here, but we're glad that you're doing well over there. I'll pass the mic to Bruno, and I think, Diego, do you have any questions that we can go right after if there's anything? I'll just pass it to Bruno right now.
Thanks, Miriam. Cisa, it's very nice to see your work, very exciting. My question is actually also regarding aging, and how autoimmunity would fit in your model, because if there is you saw an increase in TFR, and you showed that TFR actually regulated the autoantibody production. So how does this model fits in the increased incidence of autoimmunity in aged individuals?
Right. Yeah, that is a very good question. It's the kind of question that I like to investigate. So I do want to go in that direction. We do see, as I mentioned, Peter described it before, that there are more TFRs in that context, but they're less functional. So we didn't look at the generation of autoantibodies per se in this vaccination model, but it is definitely a question that I'm interested in pursuing. What else is, if these cells are not I was surprised and glad to see that they're still able to select towards the antigen, but we didn't look at what else is there, right? So that could come at a cost of also generating maybe pathogenic or more autoreactive antibodies, which we didn't look at in this model. But yeah, but it's definitely a question I'm interested in. Yeah.
Thank you. Diego, do you want to open the mic over there?
Yeah, so we didn't have questions from the students, but I have a question. Actually, I think you answered meeting already. I had a question about infectious challenge, but you mentioned you didn't do that yet. But another question I had is, since you're immunizing with you're generating anti-COVID antibodies, did you check about mucosal IgA secretion, if there is any kind of difference when you decrease the number of the TFHs or the TFR cells?
Right. Yeah. No, we didn't look at that in this paper. So we're kind of moving back to a flu infection model where we have all the tools, and it's also more related to mucosal immunity, where we can challenge the mice and we can see memory cells also in the lung and all that. So we are currently working on these other kind of questions. What I can tell you from the sequencing pipeline, we do fish some IgA cells. In this particular work, we focused on the IgG response, but our sequencing pipeline does have IgA incorporated to it as well. And every so often we do find an IgA cell, but we focused on the lymph node. So I think with the flu model, looking at lymph node and lung altogether, we might be able to find more and explore more that side of the response. Yeah.
I think we do not have any other questions around here. Do you have it there, Miriam?
Okay, thank you. I think just one more. I wonder about the adjuvant that you used. Do you plan on, or do you think it'd be relevant to try different ones, or do you think that that's already what you have been using and it is similar to what it would be used in a vaccine for humans, equivalent, not the same, but
yeah. Yeah, the idea for this study was to mimic as much as possible the clinical vaccine, and that's why we went with MF59. We do use different adjuvants in the lab, and we know that some of them give a better or worse response. So there is a role also for other molecular pattern recognition involved in this crosstalk. But yeah, we do use different adjuvants and infection versus vaccination to kind of assess that as well.
Okay, and have you tried the infection versus vaccination, the DTR mice, or does it? You haven't done that. Well, I guess not for SARS-CoV, but
yeah.
Do you see if there is a big difference in terms of epitope use, or once you have vaccinated, you do focus mostly on the epitopes that have been mostly responded against during vaccination?
Right. Yeah. We do that kind of to narrow down the analysis. We usually focus on like HA specific for flu or NP or the common well-known antigens that are targets of neutralizing antibodies. But we are using also the deleter models with flu infection. Yeah. Okay.
Okay. I think we don't have any more questions on the side here either. There is a message here from Andrea. Andrea, do you want to open your mic if you can? Should
sorry,
I read it?
I just wondering about the can you listen to me? Oh,
yes.
Excellent presentation, Cis. Thank you. Thanks a lot. It's amazing how you're progressing, and I'm so happy for that. About this secondary germinal center and the difficulty of the B cells to re-enter the germinal centers.
Mm-hmm.
We know that the TFR maybe, or the regulatory T cells, are related to the termination of the germinal center, right? But do you think these TFRs are somehow playing a role in blocking those B cells to re-enter into the germinal center? Did you check for the frequency of re-entry B cells in the secondary germinal center?
Yeah.
Or if you delete the TFRs in the secondary immunization, I don't know if you check for that.
Yeah, we did. I think maybe I put it here in the, let me see, after the end. I
Okay.
just didn't want to pack too much into the, but we did delete it, and the boost, no, it's not here. Yeah, we did delete it during the boost as well. And I do think, well, we're not sure if TFR cells are directly interacting with B cells. So how there being that bottleneck, we do think TFR cells put a bottleneck on which B cells are coming into participating in the GC, but we're not sure if that's a direct TFR B cell interaction or if that's through TFH cells. If TFR cells kind of limit TFH cells, and that's what limits which B cells come in. So that's why we're kind of looking more into the TFH compartment right now to see what are the features of these TFH cells right after participating on a GC, or do they come back, or do they never leave the B cell follicle? Are they always there, and they are the ones that are actually selecting in favor or against memory B cells? So the bottom line is we don't know, but we have all these working hypotheses of either a TFR cells interacting directly with B cells and putting this bottleneck for memory B cells to not get into the GC again, or the pool of TFH cells is more diverse or less diverse now, and that causes a different selection of which B cells come in, or just TFR promotes the plasma cell differentiation at first, and then all those B cells become plasma cells early on, because that's one piece we know, is that the role of TFR cells is more important early on. So once that crosstalk of B and TFH is happening, TFR cells have less of an effect there. So we do see that TFR cells have a role in the recruitment, so-called phase. So maybe they favor the plasma cell phase, and then once they're away from that reaction, B cells come into the to interact with TFH. So this is more or less like a few hints that we have that this might be the case, but we definitely have no proof yet of how that's happening. Yeah.
All right, thank you. Great talk.
Thank you.
So thank you very much, Cissa. Leonardo Travassos is also congratulating you on your work. I think the same message from all of us. It's very, very, very beautiful to see you growing and your career growing as well. So thank you again for sharing with us the work that you'll be doing there. We definitely will invite you again in a little
time. I'm always happy to come.
To hear the follow-up on this work.
Yeah.
So thank you very much. Thank everyone. And I think we meet again next week.
Nice. Thank you. Thank you, everyone.
Thank you, Cissa. Obrigada.